So, chapter 8, crazy chapter. I kind of soft-started you guys before this crazy few days of exams and quizzes and everything. So, few rules that I want to just, again, make sure that I review for you guys. We're going to focus on cells. Obviously, viruses and all of that are not included in this. Why? Because they use the processes of the cells. Differences between us and prokaryotes. Prokaryotes usually have one single chromosome. It's double-stranded and it's circular. They can have some external pieces of DNA called plasmids that have extra characteristics. But what lets them function, what's most important for them to replicate is this. And this is the chromosome that it's expressed mostly to allow the cell to survive. For us, our material, our genomes for eukaryotes, for us, fungi and protozoans, is in the nucleus. It is linear double-stranded DNA. Okay? Again, rules with regards to DNA. Hopefully you guys looked at the video that I have. So reminded yourself DNA is double-stranded. It's a nucleic acid molecule made up of nucleotides. Each strand is made by sugar phosphate backbones, so covalent bonds between the sugar of one nucleotide and the phosphate of the next one. How they create it is the carbon number 3 of the sugar makes a covalent bond with the phosphate of the other. So, carbon number 3 with the phosphate of the other one. Each strand has a direction based on the sugar phosphate. So you look at it from the top. If you hit the phosphate, that's connected to carbon number 5. So we say that's the 5' end. And the bottom is going to be carbon number 3. So that's 3' end. Then to make double-stranded, you put 2 strands in front of one another with the bases facing each other. And it's anti-parallel, meaning one goes 5 to 3, the other one goes 3 to 5. Complementary, A and T always combine, C and G always combine. Hydrogen bonds in the middle, so it's much easier to break that. The covalent bonds that you make for the sugar phosphate is extremely strong. So even boiling temperature points around 95 degrees just breaks the hydrogen bonds, does not break the covalent bonds, so it's very sturdy. All right, the other things that I said kind of have an understanding is, again, how they're showing you here the arrow that is pointing in a way. Basically, we have, in the process of DNA replication, I said it is semi-conservative, so meaning when you want to replicate DNA, you don't come and look at DNA and create a brand new identical one next to it. What is going to happen is you separate the strands, break the bonds. That is called unzipping. So, breaking the hydrogen bonds, that's called unzipping DNA. So that's unzipping. You are unzipping DNA. Because it looks exactly like a zipper. And you never break it all at once, you do it little by little. So you make semi-conservative, meaning new DNA has an old strand plus a new strand. Okay? When you unzip DNA, when you can see or exposing the bases, we say that's a template. So you read DNA based on the order of the bases. So you create a template for reading. The last thing I said, your DNA polymerases are your most important enzymes in the process of replication. So you have DNA polymerases, meaning an enzyme that makes DNA polymer. You have 1 through 3. Remember I said They are making gods, so they have a few requests. They are divas. First off, they can never, if they see a base, for example, an A, and they're supposed to put a complementary T in front of a T nucleotide, they can't do it because they want to have a 3' carbon here free to make a covalent bond there and put the T. So that's their request, which is very hard for the cell to do because your purpose is to make new DNA. Where are you going to get that nucleotide? Which we're going to see that. The other part is if they are making DNA, all right. So they have made this. And when they come and there was already another nucleotide or another strand of DNA there, they refuse to make that sugar phosphate covalent bond. So they will just fall off and they say, deal with that. Fix that little one. So that's DNA polymerases. Whether it's 1 or 2, sorry, I went down. So that will remain as a gap. We have to deal with that too. Right? The enzymes that are involved in the process of replication. And also remember I said DNA replication is what we do to DNA. This happens when the cell is ready to divide. In a bacteria, when the cell is reproducing, that's the only time it will divide. In us, for example, as humans, it's not just for our reproduction. You cut yourself, that dead cell needs to be replaced. So the healthy cells next to it have to divide so they can fill the gap and fill the tissue damage and fix you. So for that case, again, your cells will divide. Before any type of division, when you divide, your germ cells divide to make eggs and sperms. Before any of that happens, you have to replicate your DNA. Okay, so that's what you do to DNA to pass it to the new cells. Gene expression or protein synthesis that we look at later, that's how DNA functions. That's what DNA does. That's the effect of DNA. Okay. Enzymes responsible for DNA replication. The ones that I want you to know are first helicase. Helicase's job is unzipping, so unzips. Then we have another enzyme called primase. It makes an RNA primer. I'll tell you what that is. Then you're going to have DNA polymerase 3, making DNA. DNA polymerase 1, replacing RNA primer. Okay. And finally, DNA ligase fixes sugar phosphate backbone. Okay. To go through this process,